So I'm starting. My name is Aristotle Koutsiaris, and the title of my presentation is COVID-19 microthrombosis in vivo assessed by conjunctival video microscopy. My presentation is going to have two parts. For some, it is a little bit slow for the next slide, but it's okay. The parts of my presentation are going to be first a brief review of SARS-CoV-2 cell cycle and thrombotic action, and part two, the latest research on COVID-19 microthrombosis in humans in vivo. So we will start with the first part. As you can see here, this is a schematic diagram of the virus cell cycle inside an endothelial cell from Chilamakuri et al. 2021. And as you can see, there are 14 different steps which have to do with how the virus enters the cell, replicates its RNA, then there is the transcription and translation, and then how the nucleocapsid is formed, and then how it exits multiplied from the cell. And the interesting about this schematic is that you can see how the various antiviral drugs affect each step of this cycle. For example, hydroxychloroquine affects the fourth step, which has to do with endosome acidification, whereas the remdesivir, a very known antiviral drug, affects the eighth and ninth step, which has to do with the RNA-dependent RNA polymerase. However, when this cycle completes, what is going on is that we have an endothelial damage. This is confirmed. And then we have thrombosis, and then we have impaired vascular tone. So this is a schematic of the thrombotic action of the virus. And as you know, the injured endothelium has a central role in thrombotic microangiopathy as one of the three components of the Virchow triad. This is a hypothetical image from Huertas et al. It is early 2020, and here was a suggestion about how the virus affects the thrombotic action. The certain thing is that there is microthrombosis, as you are going to see later. The question that remains is how exactly this whole mechanism works, and how exactly we have this microthrombi inside the microvessels. And we are going to say a little more about this later. So we can proceed now to the proposed microthrombotic mechanisms. One was the endothelial glycocalyx damage. Another was endothelial cell-derived microvesicles. A third was a complement-mediated extracellular vesicle release. Another was endothelial exocytosis, neutrophil extracellular traps, and degradation of endothelial junctional proteins. All of these are proposed microthrombotic mechanisms. The essence is that regardless of the underlying endothelial mechanism, the extensive microthrombosis guided the clinical doctors to the proposition of tailored antithrombotic strategies. However, the benefit on the microcirculation of using antithrombotics is still a matter of debate, as you can read in a letter by Spiropoulos and Bonaka, Lancet 2022. Another useful recent information is by Watson et al. 2022, who reported that therapeutic efficacy of low molecular weight heparin appears blunted in COVID-19, is that COVID-19 appears to induce pathophysiological changes to clot microstructure. This supports our view that something strange is happening with COVID-19, and we are going to discuss this further later when I'm going to show you our results. So we proceed to the second part of our presentation, which is the latest research on microthrombosis in humans in vivo. So this part has to do with in vivo results, not from post-mortem dissections or something like that. And during the COVID-19 pandemic, not much has been reported on microvascular alterations during or after the COVID-19 disease in vivo. For assessing potential pharmacological benefits in the treatment of COVID-19, more quantitative data are needed from the human microcirculation in vivo. So the next question is, how are we going to gather in vivo data? And as I'm going to show you right now, there are 3 windows in the human body for non-invasive measurements in the microcirculation. First, the sublingual tissue window at the bedside with tissue contact. Second, the fingernail fold window at the bedside with tissue contact. And third, the conjunctival tissue window with no tissue contact. So there are 3 windows, and of course, in all of them, we need some kind of magnification because, as you all know, microvessels are not visible to the human eye. So we proceed with the first window. The technique is called nail fold video capillaroscopy, or NVC, and this is an image of the instrument in contact with the finger. One of the first results was by Natalilo et al. 2021 using NVC, and they reported qualitative microvascular abnormalities in COVID-19 patients with no controls. So this column here is COVID patients in the acute phase, and this column here is COVID patients in the recovered phase. So as you can see, hemosiderin deposits were statistically higher in the acute phase, and capillary density was statistically lower in the acute phase. So this was one of the first results, and it was qualitative. Using similar results, Koutsiaris et al. 2022 proposed an early and a late capillaroscopic pattern for COVID-19 disease. An early capillaroscopic pattern in acute COVID-19 patients is characterized by angiogenesis and microhemorrhages, as you can see in part A of this picture. And the late capillaroscopic pattern concerns COVID-19 survivors and is characterized by loss of capillaries, as you can see in the part B of this picture. The second window, as we have said, is the sublingual tissue window, or SVC. And this is an image of the instrument that is commonly used for this window. It has green LEDs here and here, a magnifying glass, and the CCD camera. And this is an example image of the sublingual tissue. Using this technique, SVC, Rovas et al. At 2021 reported significant reduction of blood velocity in comparison to controls. So as you can see here, the controls are in green box, and the COVID-19 patients are in orange, red box, but the difference is only from the mechanically ventilated patients, which are shown here with a red box. However, they did not discriminate diameters or venular and arteriolar side, Nevertheless, they gave very important information on biochemical markers concerning COVID-19. And we proceed to the third tissue window for the human body, which is the eye conjunctival tissue. The technique is called conjunctival video capillaroscopy, or CVC. And as you can see, the main component is the slit lamp. The slit lamp here is connected to a CCD camera, the CCD camera to a personal computer. And here is the human eye and the conjunctival. And images are driven to the personal computer for analysis.
Excuse me, Dr. Corser, the slides are not moving actually. It is stuck at one slide, only the demographic data.
Yeah.
Yeah, slides are not moving forward. I mean, I can see it is stuck at one slide only, which is showing the demographic data. So is there any issue from your side or from the conference side?
Now?
Oh, now, yeah.
Now it's better?
Yeah, it's better. It is moving. Thank you so much.
I'm continuing from where I left.
Okay, okay. Sure.
This is slide 14. Do you, maybe I should make a quick review of the slides I have shown?
No, it's okay. It's okay.
It's okay.
No. Yeah, it's
so,
okay.
This is our experimental setup, and this is the slit lamp, the CCD camera, and the personal computer. And here you can see a our high magnification, high speed slit lamp digital video microscopy. This is the CCD camera. This is the computer screen with a dedicated software for image analysis. So let's see now where we measured in our COVID-19 study. This is a diagram of the cardiovascular tree from the famous Guyton medical physiology. So we have the heart, then the aorta, large arteries, arterioles, capillaries, venules, small veins, large veins, then back to the heart, then the pulmonary circulation, then back to the heart. So our site of measurement was here in the capillaries and post-capillary venules. So we had no confounding factor from this. We had exactly a certain point of measurement in the vascular tree, and we introduced a new index for quantifying microvascular occlusion called percentage of occluded vessels, or POV. POV was defined as the ratio of the number of occluded vessels, NO, over the total number of microvessels, NT, at a given vascular field of view. POV was defined in fields comprising at least 4 microvessels in total, and the microvessel was considered as occluded when the flow inside was completely stopped or when blood velocity was less than 0.1 millimeter per second. So I'm going to give you an example of a stopped or occluded vessel. Here we have the initial image. As you can see, the black arrow points to a microvessel, which is considerably smaller than 30 microns. And this image on the left is the initial image. And after 14 frames on the right, we have another image. And as you can notice, the red blood cell column has not advanced at all. So this vessel was occluded because the blood flow was stopped. So in this way, we quantified microvascular occlusion. And now I'm going to show you some information regarding our groups. The first group was our control group. The second group was the COVID-19 group. As you can see, the variables of age, body mass index, body surface area, beats per minute, and mean pressure are all approximately the same. There is no statistically important difference. And here, the next slide is about some clinical data concerning only the COVID-19 group. And as you can see, we have some data. We have one column during hospitalization and one column after hospitalization. As you can see, during hospitalization, all patients had Horowitz index less than 300. They all had oxygen therapy. And most of them had antiviral therapy and antithrombotic prophylaxis, which is in the red box. After hospitalization, 50% of the patients continued to receive oxygen therapy and 100% continued to receive antithrombotic prophylaxis. We measured here after hospitalization, and I'm going to show you now our results. Our first impressive result was about POV, the percentage of occluded vessels. And as you can see, the gray column is for COVID-19, the white column is for controls, and the difference is huge. The average POV of the COVID-19 group was 6 times or 600% higher than that of the control group. So we had a huge difference here. And these results set serious questions regarding the efficiency of thromboprophylactic doses for COVID-19 patients at the microvascular level. Some of them had therapeutic doses with no better result. So considering that low molecular weight heparins bind to it, potentiate antithrombin to form a complex that irreversibly inactivates clotting factor 10 in the common pathway of coagulation, we have to ask ourselves if we are missing something. So, making a review of what I have said, we have the COVID-19 infection, we have hospitalization with thromboprophylaxis, we have exit with thromboprophylaxis, but microthrombosis persists. So this led us to the hypothesis that maybe an unknown coagulation factor exists with the name XC, with C from COVID. This unknown factor, XC, could be related to one of the mechanisms that were proposed as the cause of the microvascular damage, such as degradation of endothelial junctional proteins and glycocalyx, endothelial cell-derived microvesicles, and endothelial exocytosis and immunothrombosis with nets. So maybe one of these mechanisms has to do with this unknown coagulation factor. In addition, according to the classical Virchow's triad, stasis of blood flow is one of the 3 factors leading to thrombosis. Therefore, it is evident that microvascular blood flow at velocity plays a significant role in the thrombotic process. And these are our results concerning hemodynamics. The diagram on the left is for diameter, and the diagram on the right is for axial velocity. We have subdivided the microvessels in 3 groups according to their diameter. We have capillaries with the smallest diameter, post-capillary venules of size 1, and post-capillary venules of size 2. And here, the same as you see. We had to exclude the diameter as a confounding factor. So as you can see, both groups had similar diameters. But when we measure a velocity, as you can see in this diagram, the difference is very high. And COVID-19 patients had a significantly lower blood velocity in comparison to the controls. The significance level inside the red box was less than 0.001. P was less than 0.001. So this is another impressive result. And if we combine this with velocity diffusion equation, this is a relatively new equation that was proposed recently in 2023, but is based on the famous flow velocity equation. And as you can see, for each microvessel, there is a velocity at the equilibrium, which corresponds to a diffusion of substances also at the equilibrium. So when the velocity is lower than the equilibrium velocity, we have a smaller diffusion, lower diffusion of substances. And when we say substances, we include the oxygen inside this term. So this can lead us to the following suggestion and proposition. We make a review here. We have the COVID-19 infection. We have hospitalization with thromboprophylaxis. We have exit with thromboprophylaxis and severely lower blood vessel velocity. And this can be the cause of lower diffusion of important substances such as oxygen. And this could be a good probable cause for long COVID syndrome. So these were the two main results of our study. One was the increased microthrombosis, and the other one was the substantially reduced velocity inside the microvessels. This next slide is about a special issue in the Journal of Biomedicines with the title Blood Microbiology in Health and Disease, in which I have the honor of being the guest editor. So if any one of you has some work on this with this title, he could submit by July of 2023. Thank you very much for your attention.